Да. Все, я уже могу листать, да? А что-то не листается. Так, прижимаю мышью и листаю, да? А, вот как они, такие сверху вниз. Так, извините, сейчас необычно немножко. Спасибо, все, я могу начинать, да? Они уже меня слышат, так? Все, спасибо большое. Спасибо. Мгм. Good afternoon, ladies and gentlemen. Topic of today's lecture is antimicrobial drugs. This is lecture number 4, and mostly we will try to characterize so-called antibiotics. But antimicrobial drugs, this group of the medical preparation include many substances. And antimicrobial drugs may be subdivided to the 3 major groups. First, chemotherapeutic drugs. Second, bacteriophages. And third, prebiotics and abiotics. Chemotherapeutic drugs are antiseptics, disinfectants, antibiotics, and sulfonamides. Antiseptics, examples: iodine, alcohol, miramistine, hexidine. Second group, disinfectants, examples: chloramine, formaldehyde, sodium hypochlorite. Third group, antibiotics, penicillin, chloramphenicol, tetracycline, and so on. This would be the major topic of today's lecture. And the fourth subgroup of the chemotherapeutic drugs are sulfonamides, Bactrin, and other. Bacteriophages. You know that bacteriophages are viruses that parasitize the bacteria. Due to this, such bacteria, staphylococci, and some enterobacteria may be disrupted by administration of bacteriophages. And the last group for antimicrobial drugs are probiotics and abiotics. You know that probiotics and abiotics are preparations consisting of the leaf microorganisms, normal form microorganisms that are replicative in the contrast to the chemotherapeutic drugs. Bacteriophages are also replicative, but bacteriophages viruses or probiotics consist of the bacteria struggling against pathogenic bacteria. Probiotics examples: Bespharin, Lactobacterin. Bacteriophages, you know, are the viruses of bacteria. Bacteriophages may cause lytic bacterial infection, that is shown in the left part of the slide, is the 1, 2, 3, 4, 5 stages of the interaction of bacteriophages with tadpole particles, bacteriophages to the host cells that are shown by the red purple ovals. So, bacteriophages may cause lytic of bacteria. This lysis of bacteria is a result of the replication bacteriophages inside of the bacterial cells. Lytic bacteriophages may be used for laboratory diagnosis and for the therapy of bacterial infections. In the photograph, you see here a culture of the Bacillus cereus lysed by the bacteriophage that was dropped on the surface of this bacterial growth. This is a bacteriophage typing demonstration of this laboratory test. As a therapeutic mean, bacteriophages may be used for treatment of such diseases as the staphylococcal infection. What you see, staphylococcus bacteriophages. In the other photograph, this is a jar with the solution containing these viruses. Bacteriophages, exception for lytic infection, may cause lysogeny or integrative infection. Lysogeny or integrative infection is shown in the two low parts of the left slide, where you see bacteriophage injects red, black recombine genome to the bacterium, and this where the genome integrates to the chromosome of the bacteria. Integrative infection, the infection where progeny variants are not produced, but lysogenic bacteria survive and carry prophages, the parts of their genome, for example, the bacteriophage integrate into the bacterial chromosome. This is briefly repeating the bacteriophage. Probiotics and aerobiotics. They live non-pathogenic bacteria that may be effective. First, in recolonization of particular sites of the body to return the normal flora after the antibiotic therapy. In the second part of the lecture, we will see that antibiotics are chemical substances that may destroy bacteria, affecting different mechanisms, bacterial structure, physiology. After administration of antibiotics, in some cases, in the gastrointestinal system of humans, normal flora bacteria becomes killed, and this case chance for colonization of the gut by the external pathogenic bacteria is high. But in the recolonization of gastrointestinal system after the finishing of antibiotic therapy, probiotics and biotics are very important because they prevent coating of our mucous membranes by the films consisting of the pathogens that may destroy our mucous membranes. So also probiotics may be effective in enhancing colonization resistance to pathogenic bacteria. Enhancing colonization resistance means if you administer antibiotics perorally, probiotics perorally, these preparations consisting of the liver bacteria enter to the small and after that large intestine and participate in the colonization, preventing appearance in this area of these regions of the pathogenic microbes. Also, probiotics may be effective in the excluding the pathogens from the binding sites. Now, this is a function associated to the number 2, enhancing colonization resistance. And the last one, point number 4, probiotics may be effective in reducing the inflammatory response against pathogen. Inflammatory response, this is a universal protective reaction of our organism, including immune system for our organism, against any invading from the pathogens. And in some cases, inflammatory response may be exaggerated and destructive. It means our immune response, when it's developed too intensively, may in some cases cause autoimmune diseases and allergy diseases. And in this minimizing of these negative results of inflammatory reactions, probiotics and aerobiotics also may be very important. Examples of probiotics. Preparation named biosporin. Biosporin consists of leaf bacteria of 2 species, Bacillus subtilis and Bacillus licheniformis, that occur in very high concentration, 2,000 to 8,000 million cells per milliliter. These Bacillus species, Bacillus subtilis and Bacillus licheniformis, colonize gastrointestinal system of the body, first. Second, they enhance colonization resistance to pathogenic bacteria. What in the photograph, in the painting, in the right part of the slide, lower right, you see 1, 2, 3, 4, 5 pink bacteria, the rod-shaped bacteria, that secrete bactericins, substances that destroy pathogenic bacteria, shown here the gray-blue spherical microbes. Pathogenic bacteria are killed by the Bacillus subtilis and Bacillus licheniformis due to secretion by the cells of the bacteriocins, and in this way, bacteria, normal flora, part of the probiotic, destroy pathogenic bacteria and prevent the colonization of the intestinal system. Also, by aspirin excludes the pathogens from the binding sites. Of course, layer of the normal cells prevent the attachment of pathogens to the mucous membranes. And of course, effect number 4, by aspirin reduces inflammatory reactions that may be developed in the gastrointestinal tract. The next group of the antibacterial drugs is antiseptics, AS. You know that antiseptics are chemical disinfecting agents that inhibit or kill most pathogenic microorganisms with sufficiently low toxicity for host cells. It means antiseptics kill majority of pathogenic bacteria, can kill human cells, and can kill all the non-pathogenic bacteria. Antiseptics can be used directly on skin, mucous membranes, or wounds, surface of wounds. Antiseptics are less toxic than disinfectants. Explain examples of antiseptics: phenols, iodine 3%, hydrogen peroxide, alcohol 70%, ethanol, isopropyl alcohol. Cationic detergents, for example, chlorhexidine, structural formula is shown in the left part of the slide, and miramistine, discovered in our department. Structural formula of this antiseptic is shown in the right part of the slide. Disinfectants. Disinfectants are more toxic for human cells than antiseptics. Due to this, disinfectants may be used to destroy pathogenic microorganisms on the inanimate surfaces, not in the human wound or human intestinal tract, but on the surface of the desk. Where do you want to destroy bacteria in the equipment? But disinfectants are toxic for human cells. It can be used to disinfect surfaces of our body. Examples of this rather toxic for humans substances, disinfectants, are from 2 to 8% formaldehyde, sodium hypochlorite, and some other. And finally, beginning from this slide, we'll try to characterize so-called antibiotics. Classical designation of antibiotic is a chemical substance produced by microorganisms which has the capacity to inhibit the growth of bacteria and even destroy bacteria and other microorganisms in dilute solutions. This is the formula of the Salman Waxman, formulated in 1940 year. Waxman, this is a famous American microbiologist, discovered many antibiotics. For example, streptomycin, anti-tuberculosis drug. In the lower part of the slide, you see structural formulas of two families of the antibiotics. In the left part, you see formula of the penicillin and penicillin family substances, and in the right, cephalosporin and cephalosporin family substances of the first, second, third, and other generations of these antibiotics. You see that both penicillin and cephalosporin, both these types of antibiotics, have so-called beta-lactam ring here and here. Beta-lactam ring is shown by the blue color. This is a 4-component chemical group. One of the components of that is a nitrogen molecule. Beta-lactam ring is critically important for realization of the antibacterial potential of penicillins and cephalosporins. Penicillins were discovered first as antibiotics. History of the antimicrobial therapy. The first systemic attempt to find specific chemical substances to treat infectious diseases was made by the Paul Ehrlich. These scientists searched for so-called magic bullet. This is a term introduced by Paul Ehrlich. Magic bullet that would destroy bacteria without destroying of the human cells. Paul Ehrlich coined the term chemotherapy and found a salvarsan to be effective against syphilis. Salvarsan was in the 1 century before very important part of the treatment of the syphilis. This is a sexually transmitted disease. Alexander Fleming. This is American microbiologist. In 1928, Alexander Fleming observed that the antistaphylococcal effect of penicillium mold and was the first to recognize its potential for countering infections. In early 1940s, his idea came to fruition when the method of mass production of penicillin was developed. Penicillin historically was the maybe most important antibacterial drug used to treat people wounded during the Second World War. And this was the very, very important drug that helped to survive million people in this period. The third famous scientist is Herher Domach. Herher Domach in 1935 discovered the Prontosil. This is a chemical substance with the formula of sulfonamide. What in the chemical formulas below the Gerhard Domach photograph, you see substance drawn by the brown color. Well, this is a basic formula of sulfonamide, and this formula fits for the Prontosil. Prontosil, this is the red dye with a sulfonamide portion, what is the sulfonamide group, in a molecule that may inhibit growth of many gram-positive bacteria, including staphylococci. This discovery of Herher Domach stimulated the development of a group of antibacterial substances known now as sulfonamides. Sulfonamides are effective against a variety of pathogenic bacteria via prevention of folic acid synthesis. Many bacteria are capable of producing folic acid. What here in the white field in the chemical part of the slide, you see structure formula of the folic acid. For us, folic acid, this is a vitamin B9. This vitamin is the precursor of the synthesis of the adenine, guanine, cytosine, and thymine, and many amino acids. Of course, this vitamin is absolutely required for surviving of mammalian organisms and for bacterial cells also. We don't have enzymes that help to produce folic acid, but bacteria have these instruments, and what even these instruments are targets for sulfonamides. The next scientist, famous scientist, is Salman Waxman. This American scientist. The development of the antibiotics resumed with this scientist who was born in Ukraine and moved to the United States in the 1910, in the childhood. In 1940, he discovered streptomycin and defined an antibiotic as a chemical substance produced by microorganisms which has the capacity to inhibit the growth of bacteria and even destroy bacteria and other microorganisms in the dilute solutions. This is a repeating of the formula that you saw in one of the slides before. This designation of antibiotic was introduced in the scientific physiology by Waxman. Streptomycin, discovered by Waxman, constituted a major breakthrough in the treatment of tuberculosis. Really, streptomycin was the first really active antibiotic antibacterial drug capable of inhibiting replication of tuberculosis agents in the human body. In the same decade, Waxman isolated exception for streptomycin, second antibiotic chloramphenicol, the next neomycin, and chlortetracycline. General properties of antimicrobial drugs, including antibiotics. General property of antimicrobial drugs. This is a selective toxicity. Selective toxicity of antimicrobial drugs is the selective inhibition of the growth of the microorganism without damage to the host. This selective toxicity, this is a major property of the so-called magic bullet that was searched for by Paul Ehrlich. Do you remember Paul Ehrlich introduced this term, magic bullet? Why magic? Because it has selective toxicity with this capacity to damage microbe without damage to the host. This is the most important concept underlying antimicrobial therapy. Selective toxicity may be achieved by exploiting the differences between the metabolism and structure of the microorganisms. Of the microorganisms, bacteria, viruses, and fungi, and the metabolism and structure of the host human cells. Today, antimicrobial or antibacterial drugs is a special group of chemotherapeutic agents used to treat diseases caused by microbes. Often they are also called antibiotics. Antimicrobial drugs nowadays can be made from cultures of microorganisms, for example, fungi, or artificially by the chemical synthesis in the laboratories. Antimicrobial drugs can be subdivided depending on their spectrum of activity, character of activity, and mechanism of action. So, all the antibiotics may be subdivided on the basis of the spectrum, character, and mechanism of action. Spectrum of antibacterial activity. Spectrum subdivides all the drugs for broad spectrum antibiotics and narrow spectrum chemotherapeutic drugs. Broad spectrum antibiotics are active against several types of microorganisms. Narrow spectrum antibiotics are active against only one or very few types of microorganisms. Example of broad spectrum antibiotics: carbapenems, chloramphenicol, fluoroquinolones. Examples of the narrow spectrum: penicillin, glycopeptides, rifamycin. In this slide, you see spectrum of bacterial activity shown as the line overlapping more than one groups of bacteria. Here you see 1, 2, 3, 4, 5 columns of the different color characterizing different types of bacteria. What the left orange, not pink, mycobacteria. Then gram-negative bacteria shown by the blue color. The massive the gram-positive bacteria by the yellow, chlamydia green, and rickettsia purple. Broad spectrum antibiotics, for example, one of the broad spectrum is tetracycline and tetracyclines, members of this family of drugs. You see the tetracyclines may be effective against gram-negative bacteria, gram-positive bacteria, chlamydia, and rickettsia. In the contrast, polymyxins are effective only against some of the gram-negative bacteria. Isoniazid is effective only against mycobacteria. The next property is the character of activity. On the basis of character of activity, drugs may be subdivided to bacteriostatic and bactericidal. Bacteriostatic drugs cannot kill bacteria, but can prevent their replication. Bactericidal drugs, they kill bacteria. Examples of bacteriostatic antibiotics is chloramphenicol, erythromycin, clindamycin, sulfonamides. Examples of the bactericidal aminoglycosides, beta-lactam drugs, penicillins and cephalosporins, for example, vancomycin, quinolones, metronidazole. Character activity in this slide demonstrate that bactericidal drugs cause the disruption of structures of bacteria and disappearance of these bacteria. In the contrast, bacteriostatic drugs may inhibit replication of bacteria, preventing accumulation of bacteria in the human body. But this bacteria remains alive. Killing of these bacteria and elimination of this bacteria from the human body is the business of the immune system. For example, phagocytes. Here you see some black, red macrophage with the blue, with the green nucleus that engulfs bacterium treated by bacteriostatic antibiotic. It means bacteriostatic drugs may eliminate bacteria only in the association to the immune factors of the human organism. And the third important principle of the classifying of antibacterial drugs is classifying on the basis of modes of action. Modes of action subdivides all the antibacterial drugs for the drugs that have different targets in the bacterial cells. Here you see theoretical drawing of the bacterial cell. So at the top you see cell wall synthesis inhibitors. Example, beta-lactams, including penicillin, cephalosporins, carbapenems, and some other. These cell wall synthesis inhibitors prevent synthesis of the peptidoglycan in the replicating bacteria, preventing formation of the normal cell wall in the progeny cells, and as a result, the lysis in the hyperosmotic conditions. To the right of cell wall synthesis inhibitors, you see phthalate synthesis. They are antimetabolites, sulfonamides and trimethoprim. How does work these things? Phthalate synthesis inhibitors prevent chemical transformation of para-aminobenzoic acid to the dehydrofolic acid and tetrahydrofolic acid. What dehydro and tetrahydrofolic acid, they're precursors of the folic acid. And sulfonamides and trimethoprim block what this pathway of the chemical modification of precursors to the folic acid. Protein synthesis. Protein synthesis inhibitors are subdivided to inhibitors protein synthesis that act against 50S subunit of ribosome, this or orange, and 30S subunit of the ribosome. 50S subunit inhibitors bind to the 50S subunit ribosome, for example, macrolides, chloramphenicol, prevent synthesis of the protein in this way. 30S subunit inhibitors, aminoglycosides, tetracycline, for example, they also inhibit synthesis of the protein. It means process of the translation of the genes. RNA polymerase inhibitors, rifampin, they prevent transcription of DNA into the messenger RNA. Cell membrane disruptors, in the lower part of the slide, on the left part you see this square. Cell membrane disruptors destroy structure of the lipid layer of the plasma membrane, causing damage in the plasma membrane and leaking of the cell contact to the environment. Example is polymyxin. In the upper left part of the slide, you see nucleic acid synthesis inhibitors, DNA hierase inhibitor or quinolones, topoisomerase. These DNA hierase inhibitors prevent the normal superspecialization of DNA, preventing normal transcription from this DNA. Some examples of the inhibitors of synthesis of antibiotics. First group of these antibiotics are inhibitors of cell wall synthesis. At least penicillins were discovered first by Alexander Fleming, and penicillin G was the first antibiotic discovered in the history of the chemotherapy. Penicillins is a group of antibiotics taken from culture of the fungus Penicillium, or made in the laboratory. What here you see, structure of the hypha and exospores of Penicillium mold fungus. The second group of the inhibitors of synthesis is cephalosporins. Cephalosporins are the antibiotics originally derived from the fungus Cephalosporium acremonium. Cephalosporins are similar in chemical structure to penicillins. Penicillins and cephalosporins are bactericidal and act by inhibiting transpeptidases. These enzymes can catalyze the final cross-linking step in the synthesis of peptidoglycan. They kill bacteria only when they are growing. In non-growing cells, no new cross-linkages are required and penicillin is inactive. Inhibition of cell wall synthesis prevents the formation of normal mirroring of peptidoglycan structure. In this drawing, you see parallel lines of the blue color. Blue color are the bridges between muramic acid and the glucosamine, alternating groups of muramic acid glucosamine bridged by the glycoside bonds that are shown green. Shown blue. This structure is cross-linked by the red bridges. Red bridges consist of peptide, oligopeptide bridges, bonds. Penicillin binds to the variety of receptors in the bacterial cell wall and cell membrane called penicillin binding proteins. Penicillins finally cause the disruption of the peptidoglycan structure. How? Because some penicillin binding proteins are, for example, some of these proteins are enzymes named transpeptidases. They catalyze the cross-linkage step in the synthesis of peptidoglycan. This cross-linkage is shown by the red color. Changes in the penicillin binding proteins may contribute to an organism's resistance to penicillin. In the destroying of peptidoglycan are important autolytic enzymes. Autolytic enzymes are examples of the penicillin binding proteins. These enzymes, some of them are named murine hydrolysis. They cause hydrolysis of the murine. These enzymes are activated in penicillin-treated bacterial cells. Penicillin activates murine hydrolysis. They are normal enzymes of bacteria, and usually they cause hydrolysis of murine in the period of the dividing of the maternal cell to daughter cells. But if murine hydrolysis are hyperactivated by penicillin, these enzymes degrade the peptidoglycan by the damaging of the peptide bonds that cross-link the peptidoglycan structure. And only blue bridges, glycoside bridges, becomes not enough to make peptidoglycan rigid. And after that, the peptidoglycan structure becomes disrupted. Some bacteria, for example, Staphylococcus aureus, are tolerant to the action of penicillin because murine hydrolysis are not activated. This is a result of the mutation of genes responsible for murine hydrolysis synthesis, and these genes make murine hydrolysis resistance for hyperactivation by penicillin. This was one of the process of mutation responsible for antibiotic resistance of some of the bacteria. For example, some strains of the Staphylococcus aureus. In this slide, you see in the left part of the slide normal suspension of the cocci before penicillin treatment. In the right part of the slide, you see the same cells after penicillin treatment. Penicillin-treated cells die by the rupture as a result of influx of water into the high osmotic pressure interior of the bacterial cell. What is happening? Peptidoglycan is disrupted. Peptidoglycan is the substance responsible for rigidity of cell wall. A cell wall is the organelle part of the bacterial cell that prevents osmotic lysis. When peptidoglycan is disrupted, osmotic lysis is happened. What does it mean, osmotic lysis? It means that water from the external of the cell is attracted inside of the bacterial cells because concentration of the proteins, salts, sugars, and other compounds is much higher inside of bacteria compared to the external space. This influx of water causes enlargement of bacteria, finally rupture of these bacteria and death of them. Basic structure of penicillin and cephalosporin contains beta-lactam ring. What is this blue and this blue? Left beta-lactam ring is the penicillin structure, right beta-lactam ring in the cephalosporin structure. Because both families of antibiotics, penicillins and cephalosporins, contain beta-lactam rings, both these members of these families are named beta-lactam drugs. Beta-lactam drugs. These beta-lactam drugs are characterized by the presence of beta-lactam ring. An intact ring structure is essential for antibacterial activity of the antibiotics. If this ring would be chemically disrupted, destroyed, antibiotic will become non-effective. They will lack their antibacterial activity. Some bacteria develop very important, powerful instrument of the neutralization of beta-lactam drugs. Some bacteria evolutionarily develop beta-lactamase enzyme that can interact with the beta-lactam ring of the penicillins and cephalosporins and disrupt one of the chemical bonds, destroying four-component ring structure. But this destroying of the bridges between nitrogen and carbon in the beta-lactam group causes neutralization of the antibiotic antibacterial activity of the beta-lactam drugs. Beta-lactamyces are enzymes that frequently occur in the resistance plasmids of the both gram-positive, gram-negative bacteria. These plasmids code for beta-lactamase, and bacteria having these plasmids becomes resistant. Beta-lactamase also is called penicillinase. Beta-lactamase and penicillinase, they're synonyms. Modification of penicillins and cephalosporins. Modification of penicillins and cephalosporins are chemical attempts made by the scientists to make beta-lactam rings of the antibiotics non-accessible for beta-lactamases. In the left column of the formulas, you see different versions of the penicillins. The natural penicillin produced by penicillium fungi is named penicillin G. The modified penicillins, chemically modified, more resistant for beta-lactamase, are shown in the lower part of this column: methicillin, ampicillin, carbenicillin, amoxicillin. These substances are antibiotics of the second and third generation of the penicillins. Analogous process was realized concerning cephalosporin. First generation cephalosporin in the top of the right column of the formulas, and low you see second generation, third generation of the cephalosporin. Second generation cephaloxime, and third generation cephaloxime. What do you see here in the purple squares? In the purple squares, you see modified radicals or chemical groups of the penicillins and cephalosporins that prevented attack of the beta-lactamase against beta-lactam ring here and here. The effectiveness of penicillins and cephalosporins has been increased by chemical changes in the side chains. Well, these purple squares demonstrate the side chains in the side chains of the structure of the substances. Access of the enzyme to the beta-lactam ring may be blocked by modification of the side chains with the addition of large aromatic rings containing bulky methyl or ethyl groups. Now, for example, pay attention to the methicillin, the first generation, so second generation of penicillins, methicillin. You see that methicillin has the radical contain phenol group, the penicillin G, but this group is supplemented with two radicals, the methyl groups, first and second. Due to this, methicillin is named methicillin because contains these methyl groups. Ampicillin has a new nitrogen containing NH2 group. Carbenicillin contains CO sodium group, and amoxicillin contains not only NH2 group but additional oxygen bridged to the phenol ring. But all these addicts make beta-lactam ring is protected. Now, analysis process you see here for cephalosporin modification. For example, cefaclor, you see additional NH2 group. In the cephaloxime, OCH3 group. Amino group also for cephaloxime. This slide demonstrates principle of the modifying of the radicals of the basic structures of the antibiotics of the certain family of drugs chemical modifications helping to protect beta-lactam green groups of these antibiotics from contact with the beta-lactamases. The next group of the antibiotics act by inhibition of protein synthesis. Beta-lactam antibiotics act by the inhibition of synthesis of the peptide glycan of the cell wall synthesis, a second inhibitors of the protein synthesis. Several drugs inhibit protein synthesis in bacteria selectively due to the differences between bacteria and human ribosomal proteins, ribonucleic acids, and the associated enzymes. Inhibitors of protein synthesis may be subdivided into 50S subunit inhibitors and 30S subunit inhibitors. 50S subunit, this is a large subunit of ribosome. 30S subunit, the smaller part of the ribosome. Cleavage between 50S and 30S ribosome usually is occupied by the messenger RNA that moves between these subunits, attracting transport RNAs that bring to this cleft amino acids that bridge one to other, forming protein. Drugs that act on the 50S subunits are chloramphenicol, erythromycin, clindamycin, linezolid, and many others. Drugs that act on the 30S subunits are aminoglycosides, for example, streptomycin, and tetracyclines. Drugs that act on the 30S subunits. They're usually bactericidal drugs, streptomycin, structural formula shown at the upper part of the slide, gentamicin, and amikacin. Amikacin is shown in the lower part of the slide. Aminoglycosides means that substances of all these antibiotics have amino groups. Amino group is NH2. But here in the streptomycin and H2, there are many NH2 groups here. And in the amikacin, NH2 groups are also present in the formula here, here. They're bactericidal drugs. It means they kill bacteria. They're useful against many gram-negative rods and certain gram-positive bacteria. Aminoglycosides have the limitation in their use. Limitation in the aminoglycoside use are aminoglycosides are toxic, have toxic effect on the kidney, nephrotoxic kidneys of humans, and on the auditory and vestibular portion of the eighth cranial nerve. Also, aminoglycosides are poorly absorbed from the gastrointestinal tract and due to this cannot be given orally. The next limitation, they're ineffective against anaerobic bacteria. Aminoglycosides act only to the bacteria that have aerobic. And finally, aminoglycosides penetrate the spinal fluid poorly. It means they usually are not used for the treatment of the brain infections. Mode of action of aminoglycosides. First, aminoglycoside may inhibit formation of initiation complex of ribosome composed of the streptomycin-treated 30S small subunit, 50S normal subunit, and messenger RNA. To start translation, protein synthesis, ribosome must be combined from large and small subunit and attract by mRNA. If one of the subunits, for example 30S, is amino acid treated, this initiation complex is not formed, and due to this, is incapable of maintaining synthesis of protein. In some cases, synthesis of protein is initiated, but in this case, aminoglycosides cause misreading of the triplet codon of the messenger RNA. You see that messenger RNA consists of codons. For example, uracil, the adenine, uracil cytosine. What uracil cytosine must bind to the complementary triplet on the transporter RNA consisting of adenine and adenine and guanine. Misleading means that triplet codons of the mRNA bind to the incorrect triplets of the transporter RNA, and as a result, transporter RNA bring and help to help insertion of the wrong amino acids phenylalanine to the structure of the grown protein. Now, this wrong amino acid involvement in the protein synthesis may cause lack of the effectiveness of this protein. Protein becomes non-functional. In this case, phenotypic results will be membrane damage if such the protein is synthesized in the aminoglycoside treated cells in bacteria. Also, aminoglycosides may cause allergic dermatitis. And here you see 3 photographs with the red, inflamed, affected, damaged skin. This is allergic dermatitis that may be caused by streptomycin and other aminoglycoside drugs. Tetracyclines, the next group of the drugs that act against 30S ribosomal subunit. Structural formula of tetracyclines is shown in the right part of the slide. Tetra means 4. We see 4 6-component chemical circle structures that basis for the naming of this substance is the tetracycline. Tetracyclines are broad-spectrum antibiotics with a bacteriostatic activity against a variety of gram-positive, gram-negative bacteria, mycoplasms, chlamydia, and rickettsia. It means tetracycline is the typical wide-spectrum antibiotic. But these antibiotics are bacteriostatic. In the contrast to aminoglycosides, beta-lactam antibiotics, they are bactericidal. Tetracyclines are bacteriostatic. They inhibit protein synthesis by binding to the 30S ribosomes, subunit, and blockade of the transport RNA association to the messaging RNA. In general, tetracyclines have low toxicity. The selective action of tetracyclines on the bacteria is based on its greatly increased uptake into bacterial cells compared with the human cells. It means uptake of tetracyclines by bacteria is much higher than uptaken by the human cells. Tetracyclines inhibit protein synthesis by binding to the 30S ribosomal subunit, and blocking the amino acyl transferase RNA. It means tetracyclines block transport RNA entering to the initiation complex consisting of small and large subunits of ribosome and messenger RNA, blocking the transport RNA from entering the acceptor site of the ribosome. Result, blockade of the growth of the protein synthesizing in the process of the translation. Limitation is the tetracycline use. Most limitations in the tetracycline use are suppression of the normal bacterial flora and brown stain of teeth of fetus and young children. First, suppression of the normal bacterial flora of the intestinal tract, which can lead to diarrhea and overgrowth by drug-resistant parasitic bacteria and fungi. Some of these fungi have the black color, and disease, some symptoms that you see here is black hairy tongue. Black hairy tongue because this black color, the color of the hive of the fungi that are stimulated in the growth due to the killing of bacteria that usually concur to the fungi for the surface of the mucous membranes of human intestinal tract. Second, brown stain of the teeth of fetus and young children is a result of the deposition of tetracycline in developing teeth. What you see, brown stain of the teeth. Drugs that act on the 50 subunit, 50S subunit. First of all, this is chloramphenicol. Chloramphenicol, structural formula shown here, contains chlorine atoms. Chlorine atoms here and here, 2 atoms. And this is one of the bases of the naming of the chloramphenicol, the chloram, because chlorine contain antimicrobial drug. Chloramphenicol is an antibacterial and anti-rickettsial drug. It is bacteriostatic against many gram-positive and gram-negative bacteria, including anaerobic bacteria. At the same time, chloramphenicol is bactericidal against such causative agents of meningitis as Neisseria meningitidis, Haemophilus influenza, and Streptococcus pneumonia. These 3 species of bacteria are considered as the most important causative agents of bacterial meningitis in children. Chloramphenicol inhibits protein synthesis by binding to the 50S ribosomal subunit and blocking the action of the peptidyltransferase. Peptidyltransferase, this enzyme that forms the peptide bond between last amino acid in the growing protein polypeptide change and new amino acid that is delivered to the RNA by the next transport RNA. The bridge between peptide bond between these neighboring amino acids becomes blocked because peptidyltransferase activity is blocked. This prevents the synthesis of new peptide bond, and the process of the elongation protein becomes stopped. Limitation in the chloramphenicol use. Chloramphenicol is known as a highly toxic antibiotic. The most important side effect of the chloramphenicol is dose-dependent bone marrow toxicity. Bone marrow toxicity. Streptomycin, no, this is a nephrotoxic. Chloramphenicol mostly is bone marrow toxic. This drug inhibits protein synthesis in the mitochondria of human cells. These mitochondria have 50S ribosomal subunits. It means our mitochondrial RNA, mitochondrial ribosomes have 50S ribosomal subunits. Among the more serious side effects of the chloramphenicol are blood disorders such as the aplastic anemia. Aplastic anemia, this is a fall of the concentration of the red blood cells and concentration of hemoglobin in the blood. Aplastic, it means initial synthesis of the hemoglobin and red blood cells is minimized, is affected. Associated to this effect, contraindications are known. Contraindications in the chloramphenicol use. These contraindications are mild or unidentified infections, pregnancy, breastfeeding, non-allergy to this drug. All these conditions, they cause the prohibition of use of chloramphenicol in the patients with these diseases. Chloramphenicol is considered as a drug of choice only for typhoid fever. If a typhoid fever is developed in the severe variant of the clinical development. Drugs across the 50S subunits, exception are, these drugs also are macrolides. For example, erythromycin and azithromycin. Structural formulas of these macrolides antibiotics are shown here. Erythromycin in the upper right part of the slide and azithromycin in the lower part of the slide. Macrolides means something is macro. What you see here, multi-component circular structure. This is macrolide structure that is responsible for naming of these drugs. Macrolides are bacteriostatic drugs with a broad spectrum activity. The drug inhibits, drugs of this group inhibit protein synthesis by binding to the 50S subunits of ribosome and blocking the polypeptide chain elongation. Blockade of the polypeptide chain elongation. Macrolides are effective in the treatment of pulmonary infections caused by Mycoplasma, Legionella, and Chlamydia species, and gram-positive bacteria. Mycoplasma, Legionella, and Chlamydia are gram-negative. Exception for some gram-positive bacteria are also sensitive for macrolides. Erythromycin is one of the least toxic of commonly used antibiotics. But resistance usually emerges due to the modification of bacterial ribosomal RNA, which prevents binding of antibiotic. It means macrolides are characterized by the quick development of the drug resistance in the bacteria due to the modification of bacterial ribosomal RNAs. And macrolides, unfortunately, can be used in the long courses because of the therapy, because during 2 or 3 weeks, bacteria becomes adapted to these antibiotics and survive. And finally, inhibitors of the nucleic acid synthesis. Drugs that inhibit nucleic acid synthesis may be subdivided to drugs that have that affect three principal mechanisms of the synthesis of the nucleic acids. First, synthesis of DNA, preventing DNA replication. RNA synthesis, when transcription is inhibited. And the third group, this is a prevention of the synthesis of the folic acid by use of the precursors of paraminobenzoic acid, such as sulfonamides and dimetaprim. Sulfonamides. But this scheme, shown in the left part of the slide, demonstrates how paraminobenzoic acid, after that, is transformed to the dihydropteroic acid. Dihydropteroic acid, after modification, is transformed to the folic acid. After that, folic acid in the form of the dihydrofolate, DHFF dihydrofolate, is converted to tetrahydrofolate. This conversion is maintained by the enzyme dihydrofolate reductase. And after tetrahydrofolate acid, this compound is involved in the synthesis of adenine, codeine, cytosine, thymine, and some amino acids. Sulfonamides in this scheme are shown as the molecule of the brown color. You see that structurally sulfonamides very similar to the para-aminobenzoic acid. And if sulfonamides, sulfon, what? SO2NH2, what this sulfur contain group, sulfonamides, if they are involved in this biochemical process, they prevent synthesis of heteropiruvic acid and after this folic acid. In the human cells and in principle mammal cells, this process of synthesis of the folic acid is absent. We satisfy our requirements for folic acids via eating of the food contain vitamin B9. B9, this is a folic acid. Examples of the sulfonamides. Bactrim, biseptol, septrim, salazidimetoxin, salazapyridazine, streptocidum, sulginum, sulfadiazine, urosulfan, etazole, and many others. What in this new slide you see structural formulas of the sum of the sulfonylamides. No, general formula is shown in the upper left part of the slide. This general formula after modification may be converted to the white streptoside, red streptoside, norsulfazole, phthalazole, urosulfan, sulfadimazine, sulfazine, etazole, sulfacyl, sulgin, and many other sulfonamides. All of these sulfonamides are available now and may be used for treatment of the many bacterial infections. Precursor synthesis. In the upper part of the slide, you see structural formula of the folic acid. Mammalian organisms don't synthesize folic acid. That also is named with the vitamin B9. Sulfonamides don't interfere with mammalian cell metabolism, and this is a basis of the guarantee of the occurrence of the selective toxicity. Sulfonamides are effective against a broad range of gram-positive and gram-negative bacteria, such as Nocardia and Chlamydia. Exception for sulfonamides. Some other drugs, for example, trimethoprim, also has the mechanism of the prevention of the metabolism of folic acid. Trimethoprim is also effective against many gram-positive, gram-negative bacteria. This is a drug of choice for the treatment of urinary tract infections. If used together with sulfonamides, trimethoprim produces a synergetic effect. It means effect of the supporting of effect of the sulfonamides. It means administration to the treatment of the chronic uropathology of urinary tract by sulfonamides, and urosulfan, and trimethoprim, was these preparations support effect of each other, causing very intensive antibacterial effect. Trimethoprim and sulfonamides that we discussed before are associated to antimetabolites. inhibitors of the synthesis of precursors of the nucleotides and some of the amino acids. The second group of drugs that commonly are associated to the nucleic acid synthesis inhibitors are ribonucleic acid synthesis inhibitors. Drugs that inhibit ribonucleic acid synthesis are rifampin, structural formula shown here, and rifabutin, that is derivative rifampin. Rifampin is a semi-synthetic drug which binds to DNA-dependent RNA polymerase, and inhibits the initiation RNA synthesis. It's bactericidal for Mycobacterium tuberculosis and some aerobic gram-positive cocci, for example, Staphylococcus aureus. And rifampin, one of the special anti-tuberculosis drug widely used now in the treatment of this disease. Because resistance can develop rapidly due to the mutations in chromosomal genes, rifampin is usually combined with one or more other antibiotics. Rifampin can cause red man syndrome. What here you see how this disease is demonstrated. Red man syndrome occurs when colored metabolic products of the drug of the rifampin accumulate in the body and eliminated through sweat glands. Through sweat glands release red liquid, sweat, and due to this disease is named red man syndrome. The very negative component of this disease is the damage of the liver by the rifampin in the patient. Due to this, very low administration of rifampin at high dose is contraindicated because this is a nephrotoxic substance that may cause damaging of the liver. The next group of the nucleic acid inhibitors are the DNA synthesis inhibitors. They are drugs that inhibit desoxyribonucleic acid synthesis, and they are named quinolones. Quinolones are synthetic agents that inhibit bacterial DNA herase, this is enzyme, and DNA topoisomerase. Both these enzymes, DNA herase and topoisomerase, which are inhibited by quinolones, are required to form supercoiled strands of bacterial DNA into bacterial cell. The first quinolone was nalidixic acid. This is a rather low molecular weight substance, formula shown here. This nalidixic acid is now replaced by the more effective ciprofloxacin, ofloxacin, and analogous products. What formula of the ofloxacin is shown in the right lower part of the slide, and you see that in the contrast to the nalidixic acid, formula of ofloxacin contains fluorine atom. What ofloxacin, ciprofloxacin, what is FLO? This is a part of the naming of the drugs that demonstrate that these drugs contain fluorine atom. Quinolones are active against both gram-negative and gram-positive bacteria. Also, the drug resistance can develop rapidly in such bacteria the Pseudomonas species, Staphylococcus, and Enterococcus. It means disadvantage of this drug's use, quick development of resistance in some species, for example, Pseudomonas. In this slide, you see repeating of the different targets for some antibiotics. So you see that what in the left part of the slide you see rifampicin with the symbol with the inverted T letter, symbol of inhibition. Rifampicin inhibits DNA-directed RNA polymerase. DNA-directed, DNA-dependent RNA polymerase causes synthesis of messenger RNA, and rifampin prevents synthesis of messenger RNA, process of transcription. The next, quinolones, on the right to the rifampin. Quinolones inhibit activity of DNA hierase and DNA topoisomerase. You see that this enzyme causes correct spatial position of strands of DNA in the process of the separation of strands of these DNAs in the process of the transcription. As a result, transcription and post-transcription spatial configuration of the DNAs becomes incorrect. Result, inhibition of the process of the transcription. The next, cell wall synthesis inhibitors. There are many of them on the right. Upper part of the slide, you see a list of the penicillins, cephalosporins, glycopeptides, carbapenems, monobactams. All these antibiotics prevent synthesis of cell wall, and finally, cells that don't have cell wall comes ruptured by the osmotic shock. Lower than penicillins, you see sulfonamides and timetoprim that inhibit inhibitor synthesis of folic acid, and they act as antimetabolites. In the lower part of the slide, you see that oxazolidins prevent normal activity of 50S subunit of ribosomes. Protein synthesis inhibitors subdivided to 50S inhibitors and 30S inhibitors. We see that macrolides, chloramphenicol, and clindamycin prevent normal function of 50S ribosome subunit, and aminonucleic acid tetracyclines prevent normal function of 30S small ribosomal subunits. Result in both cases is the prevention of synthesis or changing of the synthesis of the polypeptides, causing lack of these polypeptides of the proteins. So, synthesis of non-functional or incorrectly functioning proteins. And the last group of antibiotics that we have to discuss. Antibiotics that cause alteration of bacterial cell membrane function. Inhibitors of cell membrane are polymyxins and bacitracin. Polymyxins are also named polymyxin E or colistin or cholestymitate sodium. Cholestymitate sodium formula is shown in the lower part of this slide. Polymyxins and bacitracin destroy the cytoplasmic membranes of the susceptible bacteria, cytoplasmic membranes. They are active against multi-drug resistant gram-negative bacteria, causing pneumonia, Pseudomonas aeruginosa, Klebsiella pneumonia, and Acinetobacter. Pay attention. Polymyxins and bacitracin are active against multi-drug resistant Pseudomonas, Klebsiella, and Acinetobacter. This is advantage of these cell membrane inhibitors. Their use is limited chiefly by the external treatment of localized infections, external otitis, eye infection, and skin infection due to the high nephrotoxicity. It means advantage of the polymyxins. They are active against bacteria that are resistant for other antibiotics. First of all, Pseudomonas aeruginosa, causing pneumonia in the immunosuppressed people. From other side, polymyxins can be injected to the human body because they're highly nephrotoxic and can be used only to local treatment of the infections localizing the surface of our body. Drug resistance. Let's discuss this effect. Drug resistance may have different mechanisms. What in this slide you see? Antibiotics are shown by the blue balls. Yellow object, this is a cell. Plasmid is shown as the small ring of yellow color, and chromosome is the circular large DNA molecule. On the top of this upper part of this slide, you see inactivation of drug by enzymes. This is one of the major type of the drug resistance. You see antibiotics enter to the cytoplasm and becomes destroyed by the drug inactivating enzymes. Enzyme now is shown as the blue ball. You know that example of this enzymes is beta-lactamase that may destroy beta-lactam penicillins and cephalosporins. Below what this drug inactivation by enzymes, you see a red receptor reacting to the blue ball antibiotic. And C mechanism of resistance shown here is modification of the drug target. Many bacteria have instruments of the accumulation of the genetic changes that cause modification of the drug targets. Well, this modification may be modification of the structure of enzymes, for example, transpeptidases. If this is my enzymes would be chemically modified, beta-lactam drugs couldn't be reactive to this changed, modified targets and couldn't realize their function. In the lower part of the slide, you see modified cell wall proteins. It means that cell wall proteins are permeases. You know that complex chemical compounds can easily enter to the bacterial cell or leave bacterial cells to enter and be removed from cell. These compounds must be transported through the porine system. These porines are named permeases. But if genes coding for permeases responsible for uptake of certain antibiotic, if these permeases are changed, they becomes blocked. They close the door for the antibiotic, and antibiotic can't be transported inside of the cell. And in the left part of the slide, you see that some antibiotics, after entering to the bacterial cell, very quickly may be removed with the activation of the drug efflux pumps. These pumps also are permeases, but specialized on the uptake of the substances by removing of the substances. If activation of drug efflux pumps is high, antibiotic immediately after entering into the bacteria becomes released from this bacteria and has no time to cause the antibacterial effect. In this table, you see brief description of the mechanisms of the mechanism of drug resistance. First, inactivation of the drug by bacterial enzyme mechanism. Bacterial enzyme. Example: beta-lactamase can cleave the beta-lactam ring of the drug, inactivating this drug. Affected drugs, beta-lactam drugs such as the penicillin and cephalosporins. Anti-drug enzymes. Second mechanism of drug resistance, modification of the drug target. Example: mutation in the certain bacterial genes may lead to the synthesis of modified targets. For example, if genes coding for DNA hearase would be modified, and DNA hearase synthesized the protein encoded by the genes would be changed. These changes in principle may make this hearase, DNA hearase, resistant for quinolones. Result: quinolones becomes non-functional, and quinolones, you see, the one of the groups of antibiotics that confirm with this mutation mechanism. Aminoglycosins, erythromycin, and quinolones may be neutralized by what modification of targets for them. Third principal mechanism of drug resistance: reduction of the permeability of drug. Permeability means process of the uptake is changed and becomes impossible. Example: mutation in genes which encode porin proteins can reduce the amount of drugs entering the bacterial cells. Examples: penicillins, affected drugs, aminoglycosins, and many others. Now let's say about genetic basis of bacterial drug resistance. Genetic basis. Resistance can be developed by three major genetic mechanisms. First, plasmid-mediated resistance, when plasmid contains resistant genes. Second, chromosome-mediated resistance, when chromosome contains resistant genes. And transposon-mediated resistance, that is not shown in this figure that you see here. First, plasmid-mediated resistance to antibiotic is the most important type for three reasons. Plasmid-mediated resistance. First, this resistance occurs in many different species, especially with gram-negative bacteria. It's typical for many species. Second, plasmids frequently mediate resistance to multiple genes. It means one resistant plasmids may carry genes for resistant against 3, 4, 10 antibiotics simultaneously. And third point, plasmids have a high rate of transfer from one cell to another, and plasmid-mediated resistance may quickly spread in the bacterial population. I'm relating that this resistance may occur in different species. Transfer of this between members of different species also is possible. And in this way, conjugation maintains not only involvement in the spread of resistance of bacteria of the same species, but of the related species also. In the contrast, chromosomal resistance is due to the spontaneous mutations in genes that code for either the target of drug or the membrane-associated transport system for the drug controlling uptake of this drug. Chromosomal resistance is less of a clinical problem than a plasmid-mediated resistance because the frequency of mutations is much lower than the frequency of the acquisition of resistance plasmids. And so transposon-mediated resistance. You know that transposons are genes that are transferred either within or between large DNA, such as the bacterial chromosome and plasmids. Transposons can jump from one cell to other, but they may jump from one plasmid to other plasmid, from plasmid to the chromosome or from chromosome to plasmid. A drug-resistant transposon, it means transposon that contains genes for antibiotic resistance, is usually composed of 3 Gs flanked on both sides by the short series of the inverted repeated DNA sequences, with one repeated sequence and second repeated sequence, one repeated sequence and second repeated sequence. With this transposon, the structured plasmid is shown as the central element in the upper part of this plasmid. These inverted DNA sequences mediate the insertion of the transposon into the larger DNA, or in the larger DNA. Transposons, the 3 genes code, at least they have at least 3 genes that code for transposase. This is a gene coding for enzyme that catalyze excision and reintegration of this transposon to the other larger DNA molecule. Second, transposon usually has a repressor component that regulates synthesis of the transposase of the enzyme responsible for jumping of this transposon genetic block to the other part of the larger DNA. And third, very important for us component of transposon, this is drug resistance genes. They may be different in different transposons. And they code for antibacterial enzymes, beta-lactamases or carbapinamases or other. The use of double antibiotic therapy to eradicate resistant stage infections is one of the strategy of bypassing of the bacterial drug resistance. Now, for example, in the left part of this figure, you see cocci, for example, staphylococci having beta-lactamase attack beta-lactam ring of the penicillin and cephalosporin. As a result, this ring becomes cleaved, and antibacterial activity of antibacterial disappears. Bacteria replicate and occupy the area in the human body. Strategy, one of the strategies of the struggling against this bacteria. Strategy based on the chemical combine of the beta-lactam ring, beta-lactam antibiotic shown red, and the quinolone, for example, shown blue. What is happening? If beta-lactamase of staphylococci makes penicillin inactive due to the cleaving of the beta-lactam ring, in the same time, quinolone becomes activated, and activated quinolone causes the death of the bacteria. They inactivating beta-lactam drug at the same time stimulate release of the active quinolone, causing damage in the bacteria. It means all this if bacteria are sensitive to penicillin destroys them, quinolones remains inactive. Antibiotic sensitivity testing. Microorganisms vary in their susceptibility to different antibacterial drugs, and this susceptibility changes over time. Usually, laboratory tests are needed to show which antibiotic kills the microorganism selected from a particular patient. Two principal methods are in common use of the analysis of the antibiotic susceptibility. One axis is named Dix diffusion test. Second axis, dilution test. In the Dix diffusion test, pure culture of microorganisms selected from the patient are inoculated to the surface of the petri dish. After that, over this bacterial culture, you put discs soaked with different antibiotics marked by different colors, the other symbols. After that, you incubate this petri dish in the incubator at 35 Celsius during 20 hours. After that, you see that some antibiotic that diffuse from these discs cause the transparent area, the area of the inhibition of the growth of the bacteria. This area of inhibition of the growth may be measured in the millimeters, and comparing of these zones to the standard zones of the inhibition of antibiotic by this inhibition of bacteria by this antibiotic may be used to say what the antibiotics are active, or this, and this, or what the antibiotics are not active. This is a diffusion test that is not quantitative. This is qualitative test. Dilution tests are quantitative tests. They are based on the 2 times dilutions of the antibiotics in the tubes. You make the blinded sealed dilutions of antibiotic. After that, in the growth media. After that, you inoculate to every tube the equal amount of the bacteria shown red. After incubate these tubes and analyze results. Results show that with a certain concentration of antibiotic, visible growth of bacteria is finished. And this lowest concentration of antibiotic that prevents visible growth of bacteria is named minimal inhibitory concentration, MIC. What in this our example, this is a concentration achieved in the tube number 1, 2, 3, 4, 5 with a concentration of antibiotic 4 microgram per milliliter. No, start concentration was 64. What is this? The last tube 4. The next dilution in the tube number 2 will demonstrate growth of bacteria visible. It means this is an example of very, very low concentration of antibiotic. The next characteristic of the antibacterial result is counting of the relation of the minimum bactericidal concentration. How is measured this parameter? You take loopful amounts of the bacteria from all the dilutions of antibiotic that demonstrate higher concentration of antibiotic, higher than minimum inhibitory concentration. These loopful amounts of the bacterial culture are inoculated to the petri dish with the agar free from the antibiotic. You see that in the different parts of this petri dish, you will see different intensiveness of growth of the colonies. What in the tube with the 8 microgram per milliliter of antibiotic, many bacterial colonies are formed from the agar. In the case of 16 microgram per milliliter, you see the growth is less concentrated, but it's present. And in the tube with 32 microgram per milliliter of antibiotic, bacterial growth is absent. For this 32 microgram per milliliter would be minimum bactericidal concentration. Means minimum concentration that not only stop the growth of bacteria, the MIC, but kill this bacteria. This diffusion method also is named the Kirby-Bauer test. In this method, discs impregnated with various antibiotics are placed on the surface of an agar plate that has been inoculated with the organism isolated from the patient. After incubation in 35 Celsius for from 18 to 20 hours, during which time the drug diffuses outwards from the disc, the diameter of zone of inhibition of growth is determined. You see here transparent zones of the different diameter surrounding the 1, 2, 3, 4, 5 discs impregnated with the different antibiotics. The size of the zone of inhibition is compared with standards to determine the sensitivity of the agonist to the drug. The dilution method. Inoculation. The agonist isolated from the patient into a series of two-fold dilutions of the drugs. We talked about this. This is a repeat. After incubation at 35 Celsius for 18 to 20 hours, the lowest concentration of the drug that prevents visible growth of the agonist is designated as the minimum inhibitory concentration. What do you see here in the left part of the slide? You see drawing of the plate, of the plastic plate. In the right part of the slide, you see plastic plate, real plastic plate with the orange and yellow color of the growth medium. But in the left part, you see that negative control 1, 2, 3, 4, 5, 6. The blue ring demonstrated that these files, these U-bottom plastic plates, don't demonstrate bacteria because they were absent here. But in the penicillin, you see that bacterial growth in the form of the pellets consisting of bacterial culture. Again, 1, 2, 3, 4, 5 tubes, and bacterial growth is absent in the 1, 2, 3, 4, 5, 6 dilution. It means with 1 to 2 dilution of penicillin demonstrates that penicillin is not active. This is resistant from penicillin strain. Cephalosporin, methicillin, and ampicillin demonstrate lower minimum inhibitory concentrations. What is about plastic plate? Photograph, the photograph of that is see in the right part. We see that bacterial growth is shown the orange color of the medium. The plate vials are yellow, bright yellow, demonstrate no growth of bacteria. It means growth medium has yellow color, and orange demonstrate that bacterial was accumulated. It means minimum inhibitory concentration. This is a concentration of antibacterial drug minimum, causing inhibition of the visible growth of bacteria here, 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 and here. And minimum bactericidal concentration, MBC, is defined as the lowest concentration of drug that kills the organism. It's determined by taking a small sample from 0.01 to 0.1 milliliter from the tubes used for the minimum inhibitory concentration, MIC, what you see, minimum inhibitory concentration with 4 microgram per milliliter. Let's say, and spreading it over the surface. It means, again, minimum bactericidal concentration is defined as the lowest concentration of drugs that kill the organism. It is defined by the taking of small sample from the tubes used for the minimum inhibitory concentration, MIC, say, and spreading it over the surface of a drug-free blood agar plate. After incubation, the standard condition, the lowest concentration that has reduced the number of colonies by 99.9%, and really they're absent of colonies, compared with the drug-free control is the minimum bactericidal concentration. In our example, minimum bactericidal concentration is 32 microgram per milliliter. Bactericidal drugs usually have a minimum bactericidal concentration equal or very similar to the minimum inhibitory concentration. Bactericidal drugs usually have a minimum bactericidal concentration significantly higher than the MIC. What do you see here? MIC, minimum inhibitory concentration, is 4 microgram per milliliter. Minimum bactericidal concentration is 32. 32, this is 8 times higher than MIC. No, usually the if drug is bacteriostatic, difference between MIC and M by C must be more than 10. In this case, lower than 10, it means that this is a similar concentration, 432, and antibiotic shown here may be considered as the bactericidal. And the last slide, only for better recognizing of the result. Here you see serial dilutions of antibiotics. Minimum bactericidal concentration, minimum inhibitory concentration is 3.1 microgram per milliliter, because the next 1.5, 0.75, and other demonstrate appearance of bacterial growth. Minimum bactericidal concentration, where do you take from the last 3 tubes? Look for mouse bacteria and put to the agar. You see that 3.1 minimum inhibitory concentration demonstrates the intestinal growth of bacteria. 6.2 only on few colony secure, and 12.5 no one. We can consider that what 6.2 would be minimum bactericidal concentration. Now the time of the lecture is finished. I hope this lecture will help you to understand this topic better. Goodbye.